So hello, my name's Nick Holliday. I'm Chief Scientific Officer at Excellerate Bioscience and also Associate Professor at the University of Nottingham. So I have a long background in sort of molecular and cellular pharmacology and an interest really in terms of preclinical drug discovery assays. So Excellerate was founded really to provide a centre of excellence for drug discovery pharmacology. So we're a contract research organisation working across a wide range of different drug target modalities and types of drugs in terms of the way in which they work, small molecules, biologics, different mechanisms. And we look at different phases of the preclinical drug discovery cascade. But really, and alongside our lead optimization, sort of mechanism of action for us is a really important element that we bring to our clients. So I think across the types of assays that we either run or develop, understanding protein-ligand interactions or understanding protein-protein interactions is becoming really important. We have, alongside more traditional drug discovery routes, we've got lots of elements where we're looking at signaling complexes or kinetics, where we need really high-quality assays that report those protein-protein interactions or how the drugs are working at receptors or how those receptors are signaling. And fundamentally, we are really interested in fluorescence-based and luminescence-based technologies to do that. And so really our BMG plate readers, we have two PHERAstars and a PHERAstar. They're really the mainstay of the operation of a lot of our different types of assays, ranging from TR-FRET to Brett-based approaches or fluorescence, other types of fluorescent technologies as well. And we think they're fantastic in terms of being able to deliver the sort of quality data that we need actually in terms of our clients, in terms of the assay performance and getting good compound pharmacology out at the end of it. We, particularly in terms of the protein-protein interactions and also in terms of fluorescent ligand technologies, we're really exploiting these to get new information out of compound development. So we're trying, for example, to understand the way in which the kinetics of compound binding actually influences what the pharmacology will be, and actually use that to direct better compound profiling in the future. And for that, one of our real preferred methodologies is TR-FRET, time-resolved FRET. And actually, particularly the qualities of the PHERAstar FSX plate reader really fit our needs in terms of how to run those assays and how to understand the way in which these fluorescent ligands bind and how other ligands interact with them. And there are a number of things that I think set the PHERAstar apart in terms of running those types of technologies and those types of kinetic approaches. The first thing is that actually it's relatively straightforward using these techniques to get some sort of kinetics out. But in order to get really good kinetic parameters to understand our compound profiling, we actually need really high quality data. And what I mean by that is the signal to noise, the read frequency, the sort of depth of information we've got across the time course, and also kind of the ability to read, for example, long term and at physiological temperature. So if you look at what the PHERAstar gives us, the FSX that we use, for example, has laser excitation, and you might not think that's particularly important compared to lamp-based methods, but actually what it allows us to do is to use just one flash on the laser, for example, to get really good quality data out, and that then increases our read frequency. So you can imagine that quite often in terms of the kinetic analysis we want, but the first few seconds and the depth of read frequency that we get in those traces is really important for the quality data, and the PHERAstar is able to do that. It can do that at 37 degrees at physiological temperature. That's the temperature we want to look at in terms of the kinetic profiling for their compounds. The other thing that's really critical as well is that actually technically to get really good kinetics out of these assays, we actually sometimes have to work with fluorescent probes that are actually relatively low affinity. And that means much of the information comes out of that initial few seconds after we start the assay. And for that, what's really critical is a really good injector system, because that means we can start the assay through the injection and then read on a sort of flexible basis, well by well or row by row, collecting the data, almost immediately after injection, if not very, very close. And that gives us, again, data that makes us a real difference in terms of whether we get a good kinetics experiment out or one that actually is almost unusable in terms of being able to get those parameters. So currently at Excellerate we are based in Nottingham. We've just moved to a new site and a new building, so we're expanding currently. We're around about 25 staff. We will hopefully be increasing that in the future. So essentially what we want to try and do is build our capabilities, both in terms of new plate reader approaches that are coming, which I'm sure BMG will be involved with, but also in terms of other technologies as well, alongside that sort of core delivery, if you like, of really good CRO in vitro pharmacology services.